The columnaris disease is caused by the aerobic, gliding gram-negative lung rod-shaped aquatic bacterium Flavobacterium columnare. It came from the family of Flavobacteriaceae, which is composed of environmental bacteria. Below is an image of Flavobacterium columnare in two colony morphotypes, an ancestral rhizoid colony and a page-resistant rough colony. Transmission: According to Declercq et al. 2013, gills are the main source of release of the pathogenic bacteria. Also, dead fish has higher chances of transmitting the disease than a living one. Symptoms include white or grayish spots or patches on the head, fins, or gills of the fish, lesions on the back that may extend down the sides, moldy or cochineal-looking lesions on the mouth, and frayed fins. These discolored gray spots may lead to gill rot. For the methods of diagnosis, we have three: isolation, where samples were collected and incubated to see the growth of the bacteria. The other methods are enzyme-linked immunoabsorbent assay and fluorescent antibody test. External infections could be treated with antibiotics, chemicals in the water, or both. Different substances may be used in the water to treat columnaris such as copper sulfate, acriflavine, furan, and terramycin. Terramycin has proven to be quite effective both as bath and when used to treat foods for internal infections. Salt may also be added to the water to reduce osmotic stress on fish from the damage to the fish epithelium caused by the bacteria, although caution should be observed when treating other fish that are freshwater when using the salt method, because adding too much salt may cause more harm than good.
Flavobacterium columnare or F. columnare, the causative agent of columnaris disease, belongs to the family of Flavobacteriaceae. Columnaris disease was first described by Davis among warm water fishes from the Mississippi River. Although unsuccessful in cultivating the etiological agent, Davis described the disease and reported large numbers of slender motile bacteria present in the lesions. Over the decades, the taxonomic status of the pathogen has changed several times since the pioneering work of Davis. Also, they have a life cycle composed of vegetative cells, microcyst or resting cells, and fruiting bodies, or only vegetative and microcyst. Ordle and Walker reported the Myxobacterium from the columnaris disease produced both fruiting bodies and microcysts and named the organism Chondrococcus columnaris. Carnes-Job assigned the bacterium to the family Cytophagaceae and Cytophaga columnaris after isolating a pathogenic bacterium resembling Chondrococcus columnaris morphologically, but not producing microcysts. Bernardet and Grimont reclassified the organism and placed it in a family of Cytophagaceae and the genus Flexibacter, assigning it as a Flexibacter columnaris. Finally, in 1996, the bacterium receives its current name, Flavobacterium columnare, based on the DNA and rRNA hybridization data and protein and fatty acid profiles. In 1999, the F. columnare cluster was subdivided into three genome overs based on the differences in 16S RNA sequence and RFLP and the DNA-DNA hybridization.
Let us now move to the biochemical mechanism of infection. This bacteria is identified to be gram-negative that infects numerous fish species. The infection predominantly affects the external surface of the fish, including its gills, skin, and fins, as well as lethargy, loss of appetite, and gill necrosis. F. columnaris is known to survive in sterile mud or water. Its survival principally depends on the temperature, water hardness, and pH. Becker and Fujihara from 1978 reported that when F. columnaris cells were seeded in sterile mud for 77 hours, 62% survived at N Celsius, but only 35% at 20 Celsius. Fijian from 1968 reported that low pH, soft water, and low organic content decreased the survival of F. columnaris. The fish serves as the primary reservoir, and the bacteria are shed from lesions or from gills-only bonds by some fish, but several times by others. High temperatures and densities favor increased release of bacteria. In an experiment, clinical signs of disease were observed at 72 HPI, manifesting as lethargy, skin necrosis, fin erosion, and gill discoloration. In experiment number 2, it was revealed that the functioning of the branchial phosphoryl transfer network impaired in silver catfish suggests disruption of branchial bioenergetic homeostasis through failure of communication between sites of ATP generation and ATP use. The possible decrement of ATP content due to the disrupted phosphoryl transfer network induced an inhibition of ATP-dependent enzymes such as the potassium, sodium ATPase that contribute to disease pathophysiology. This study reveals that both mitochondrial and cytosolic CK activities were inhibited by F. columnare, suggesting a disequilibrium in the ATP or ADP and PCR or CR rates that may possibly lead to decreased availability of AT and disruption of the communication between sites of ATP generation or the mitochondria and ATP consumption or the cytosol.
On this part of our presentation, I'm going to talk about the scientific research and breakthroughs to control the disease, where according to the research of Laanto et al., the increasing rates of antimicrobial resistance in common disease-causing bacteria in all parts of the world were reported by the World Health Organization in 2014. Since aquaculture is the rapidly growing industry in the animal protein production, throughout the world, new methods are required for handling and management of bacterial diseases, wherein it includes one option, which is phage therapy, that is also considered a practical and efficient substitute for antibiotics in livestock, healthcare, and crop production. One significant issue in freshwater fish farming in the world is the Columnaris disease, which is currently treated with antibiotics. The disease is considered to be a good candidate for the target of phage therapy because this disease is mainly external to the fish that can be seen in the gills, fins, and skin. The first genome of phage-infecting Columnaris FCL2 were sequenced and marked for the ability to increase the survival of fish in the presence of pathogens. In utilizing the FCL2, it shows that phages have outstanding potential for controlling the columnaris disease in small-scale experiments that is also applicable in rainbow trout populations in flow-through tanks. Therefore, the phage therapy is a sustainable and efficient disease management method to the case of columnaris disease and aquaculture.
We all know that fish provides essential sources for human health, such as fatty acids and proteins. Now that we have gained awareness and insights about what may happen when our fish is infected by F. columnare, let us now discuss the final takeaways as well as how we could prevent this from happening. Columnaris is one of the most common infectious bacterial diseases in freshwater fish that may result in acute or chronic infections that typically affects the gills, skin, and fins. This infection can spread quickly and cause high mortality rates. Ecological changes and technological innovations such as the use of intensive aquaculture practices and high stock density in fish production affect pathogen epidemiology and evolution that may trigger the emergence of novel diseases. Additionally, intensive fish farming will lead to the evolution of more virulent pathogens. F. columnare can transmit efficiently from dead fish and maintain infectivity in sterilized water for months, significantly lowering the fitness cost of host death likely experienced by the pathogen in nature. Besides further elucidating the pathogenesis of columnaris disease, the development of effective measures also needs to continue. So how can the columnaris disease be prevented? Management and maintenance of a high-quality environment is of utmost importance in the prevention of columnaris disease. According to Cunningham et al., production variables such as pond depth, ammonia levels, and feed consumption were associated with columnaris disease outbreaks. This may also help identify at-risk aquaculture prior to disease outbreaks if monitored. Avoidance of exposure to the disease may also be accomplished by using UV disinfected water supplies. Studies have shown that vaccines such as aquavocol, chemotherapeutics, and probiotics may have a promising future in disease prevention and control. Reduction of fish density could also be used in the prevention of columnaris disease, especially if water temperature is high, because a lower rearing density can also decrease the transmission of ectoparasites and penetrating endoparasites. It could be an efficient tool in ecological disease management.